Great. Okay. Engineers make great, I think, make great physicians. Actually, when I got the invitation to make this talk, this is actually an email that I had just gotten in the email. So it was literally, you know, where cancer research meets engineering, right? Engineers, you're trained to kind of solve problems, right? And you have, I mean, who hasn't heard like MATLAB or looking at individual pieces? And it really gives you, when you're working with other, you know, more biology trained, chemistry trained, it really makes a nice team effort to really, you know, tackle these very difficult problems we have in medicine, you know, across the board. So kudos to all you guys who are engineers, former engineers, or just in general pursuing, you know, a career in medicine. So what are we going to talk about today? We have a vast array of audience, you know, pre-medical, medical students, some that are currently in their IR training. We're going to talk a little bit about just what is radiology, what is diagnostic radiology. I'll talk about what is vascular and interventional radiology, including in that a brief history of vascular interventional radiology and its roots and spirits in innovation. Then we'll do a lightning overview of what me as a clinical interventional radiologist does today and some of the forthcoming things that we very well may do tomorrow, as well as the changing paradigm in VIR training. So what historically, how IRs were trained, where they currently stand for those, you know, getting geared up to apply for medical school or currently in medical school. So you kind of have a sense of how do I become a radiologist? How do I become an interventional radiologist? Okay. So what is radiology? So diagnostic radiology is the use of imaging to diagnose and possibly treat disease, okay? A large part of a radiologist's training is to become the expert of the imaging, understanding the physics behind that image acquisition, as well as how to optimize that imaging. So working with other physicians, what is the best diagnostic imaging study to either evaluate for or to see how a disease condition is affecting that patient. Ultrasound up here in the top right. Can you guys see my mouse pointer? So in the top right here, this is the image of the physics of an ultrasound transducer, and this is just an example of a paracentesis. This bright white tip, that's the tip of a needle. This dark area, that's fluid that you can see nicely under ultrasound. This gray, this is an organ, this is the liver. You want to make sure you're using the image guidance to do a procedure to get into the fluid while not hitting the liver, okay? In the second row here, this is the CT, the physics behind obtaining a CT study, okay? And here is just showing you an example. You can see this is the left kidney compared to what a normal kidney looks like. There's a tumor here, all right? And then, so this is the diagnostic radiology aspect, and then talking about the interventional radiology aspect. Here, we'll talk in a bit also about ablation probes, but we're actually treating the kidney cancer that we diagnosed on the imaging study. And then on the bottom here, this is just a very, you know, basics of MRI physics, right? There's a large bore magnet, there's changing magnets that gives you this image. And on the bottom right here, this is an MR-guided breast lesion biopsy. So the diagnostic radiology is really understanding the nuances, the physics behind the image acquisition, as well as interpreting the images. So you're able to diagnose a variety of conditions. What makes diagnostic radiology so interesting, but also difficult, is you have to know what everything looks like, and you have to know the management of the conditions you see on imaging. As an example, we'll talk about colorectal cancer. All right, let's say a patient had been screened and they were found to have a colon cancer or a nodule. So that patient is probably going to have some sort of screening imaging, whether it's a CT scan, a positron emitting a PET CT scan. Well, I have to know the pattern of spread of colorectal cancer. I have to know the regional lymph nodes around the rectum. Usually those lymph nodes drain into either the portal venous system. Colorectal cancer metastases like to go to the liver, not infrequently. I also have to know what is going to make that patient a surgical candidate or not. So I'm looking at that picture and I'm saying, oh, this, this tumor has spread beyond the wall. This patient may end up getting chemotherapy and radiation before a surgeon would take them to the OR. So you're almost talking to a surgeon, you're talking to a medical oncologist based on your interpretation of that report. So you have to know everything about colorectal cancer from an imaging standpoint, as well as each individual practitioner's what the answers they want to know from the imaging. Okay. So one of the positives and negatives about pure diagnostic radiology is there's usually less direct patient contact in diagnostic radiology. Okay. This is indifferent to, we'll talk about vascular and interventional radiology in a bit here, but for the most part, a lot of diagnostic radiology, there's less patient contact. Some exceptions are if you are doing biopsies, or if you're a breast imager, a lot of times you're doing ultrasound guided biopsies on patients with lidocaine, they're awake. But otherwise, there's more limited patient interactions with pure diagnostic radiology. Okay. So we talked a little bit about vascular and interventional, about diagnostic radiology. Now we're going to transition into vascular and interventional radiology. So vascular and interventional radiology is concentrating more so on the treatment aspect of using the imaging to perform minimally invasive procedures. You're trained as a diagnostic radiologist, you're board certified as a diagnostic radiologist, but now you're doing more imaging and procedures, as well as more interaction with patients. Just some brief examples, and we'll go through much more specifics later. So doing biopsies, angiogram. So the prefix angio means vascular. So whether you're looking at the arteries, the veins, or the lymphatic channels, doing diagnostic angiograms or venograms to evaluate for conditions. And then therapeutic, we'll talk more about the procedures. So one of the main imaging modalities that we use is fluoroscopy. Basically, what fluoroscopy is, instead of a single X-ray image, it's a video of X-rays, all right? Usually you're injecting some sort of contrast agent so you can see the vascular anatomy better, and you're able to visualize structures as they move or as contrast goes through. So if we're doing a procedure with wires and catheters, we're able to see where the wire goes through while in real time we're monitoring that video of X-rays. On the right here, this is a single imaging projection of an abdominal aortogram. So here you can see the renal arteries. This is the abdominal aorta. These are some of the lumbar arteries, and these are getting into the arteries that feed the pelvis and the lower extremities. This is a single image that was taken from a fluoroscopic angiogram. To give you an example of an angiogram for a GI bleed case that we did, what you'll see here is we use fluoroscopy to get a wire and catheter into the superior mesenteric artery. That's one of the branches of the mid-aorta that feeds the large amount of the bowel. This patient had a GI bleed. And what you'll see is you'll see the normal contrast extending from the SMA as it reaches multiple loops of bowel. You'll actually see the normal appearance of what bowel opacification should look like. As the contrast goes through those blood vessels and into the venous system, you'll see the venous drainage. And in the top left of the screen here, the top right of the patient, you'll actually see the area of bleed that was eventually treated. Okay, so this is playing out now. So here's normal blood flow, the outline of the bowel here, and up here you see this contrast is still staying there. That's where the bleed was. All right, so to give you a brief history of vascular and interventional radiology, Charlie Dotter, he was born in 1920 and he was trained as a diagnostic and vascular radiologist. He was something of a radical when it came to doing things and being willing to try things. It made a lot of people nervous, but it created the specialty that you now have as becoming more and more popular. So diagnostic angiography was in the 20s. The first, Dr. Dotter did the first transluminal angioplasty in an 82-year-old woman in 1964. And this was really the the birth of interventional radiology. Basically, the patient, this is her foot, in the top left, she had this gangrenous area, and at the time, a surgeon was offering amputation. Well, the patient did not want an amputation, so Dr. Dotter said, hey, well, all right, I'm going to try to do this, basically try to do this procedure. So on the bottom left here, you can see this high-grade narrowing in her artery. Dr. Dotter performed angioplasty. You can see the artery has opened up here. This is the area that healed, and she ended up living and did not have to have her foot amputated at all. So this was back in 1964. All right. To give you a better sense of Dr. Dotter, I'm gonna play, this is actually a video that he made shortly after, a shortened version of it, to give you a sense of his personality. You guys can hear okay?
This man is in pain.
And again, his angioplasty, this is an example. Again, this was made in the 70s. It really has transformed medicine.
Intermittent claudication. His arteriogram shows a sharp, localized narrowing in the left superficial femoral artery. Atheromatous obstruction. Vascular surgery could remove or bypass it, but there ought to be a better approach, and there is. Transluminal angioplasty. Here's how it went to the man in the street. The needs of transluminal angioplasty are best met by the facilities and personnel of a modern vascular catheterization laboratory.
This is not so modern
catheter
anymore.
clots, a small quantity of heparin is drawn up for addition to the normal saline used for rinsing and intermittent irrigation during the procedure. A low-cost, disposable catheter dilating set is commercially available. This contains a coil spring guide, a number 8 inner catheter of radiopaque Teflon, a number 12 outer dilating catheter. These go together coaxially. Catheter tips are cut at an angle to facilitate entry and minimize plowing. While plethysmographic monitoring is of interest, fluoroscopic monitoring is absolutely essential to the safety and success of intraluminal manipulation. All right, let's get started. The local. A nick in the skin. Now a Seldinger needle pointed downstream. There. In with the guide and off with the needle. The smaller number 8 catheters slip down the guide and into the artery. Good contrast visualization is obtained with the use of 76% meglumine diatrozoate diluted in an equal volume of normal saline. Injections of dilute contrast agent are done whenever needed for fluoroscopic control over the procedure. Here, the first look shows the stenosis. Now to thread the guide through it, always under fluoroscopy and with care. The patient watches. This time it practically fell through. It's not always this easy. Now the smaller inner catheter goes in and is advanced over the guide and down through the narrowing. Usually there's little resistance. Next, the large outer catheter is carefully put on over the first catheter so as to avoid deforming its tip. It is then slid in and with occasional rotation to avoid plowing, is forcibly pushed through the narrowed segment.
And you guys will notice this patient is awake watching his own procedure as it's being performed.
This is also monitored fluoroscopically. This done, the actual dilatation is finished. The catheters are then pulled up above the lesion and contrast agent is injected to allow a fluoroscopic look at the results. The stenosis is gone. Before removing the catheter, a post-dilatation arteriogram is made. Increased blood flow calls for a more rapid injection. Finally, 5 cc's of concentrated heparin and a saline chaser are injected into the treated artery, and the last catheter is pulled out. Everyone was pleased. Non-obstructing manual pressure directly over the puncture site is patiently maintained until hemostasis is obtained, usually in 10 or 15 minutes.
It looks like we have it made.
It looks good, but let's not count our chickens for another day or so.
He had a right to be pleased. There being no apparent leakage from the puncture site, a non-occlusive dressing is applied. That's it. Little under 45 minutes. Next day, he's on his feet.
3 times around and still no pain. Looks like my ream job's working.
Best part right here. All right, so that gives you guys just a sense of Charlie Dotter's innovativeness as well as his kind of eccentric personality. Now I'm just in 5 minutes, real quick, we're just going to go through a lightning round of all the things where IR has innovated and how we treat various diseases. Always evaluating the patient beforehand to make sure that, you know, this is an appropriate course of action. So for cancer, biopsying cancer is obviously something we do every day. Treating cancer, there's catheter-based embolizations. So this is an example of a liver cancer where you can see these tiny beads. Either embolization is the intentional blocking of vessels. So in this case, blocking the blood vessels that go to the tumor with either, you know, various different embolization agents or needle-based ablation. Here in the bottom right, these are needle-based ablation probes that by putting, you know, either one or multiple of these probes within the tumor lesion, you're able to completely eradicate the tumor with, again, through a needle hole, all right? Talking about liver disease, hepatobiliary disease, the biliary structures, or enteral disease. On the top here, this is just showing an example of patients that are malnourished, or if they've had stroke, they need to receive nutrition, usually through a tube that's placed directly into their stomach. So this is a very common procedure we do for enteral access. In the bottom left here, this is a schematic of a stent placed. This patient has a pancreatic tumor that's blocking their biliary duct. So the bile backs up into the liver, and then the liver can't drain the bile appropriately, which causes a lot of problems. So putting a stent in to help the bile flow through. And in the bottom right here, one of the newer procedures coming out is these are multiple stones, both within the gallbladder and the common bile duct, through a needle access in the gallbladder, using a camera to look at the stones and break up the stones. To basically get rid of the patient's gallbladder and gallstone disease, again, all through a needle hole. Talking about musculoskeletal disease, and the bottom left here, this is an example of a patient that had a vertebral compression fracture. They usually come in with excruciating pain. If they're a good candidate, you go through posteriorly through the needle and either using a balloon, or these are a newer device, a spine jack device, which basically is almost like a car jack, lifts the edges of the bones and then placing cement. The vast majority of the time, the patient's pain is gone at the conclusion of the procedure by putting that cement to stabilize the fracture. So musculoskeletal disease, so we've talked about oncology, we've talked about hepatobiliary disease, we've talked about musculoskeletal disease, hemodialysis interventions. This in the top right, this is an example of a patient that had a arterial venous fistula placed for hemodialysis. And the top, this is an old abandoned system. This is contrast injection shown that the access is open. These thrombose, these are in the bottom left, these are devices used to break up clots along with medication. And now, as far as forward, we are actually starting to create arterial venous fistulas in patients needed for hemodialysis using 2 new devices. So what historically was an open surgical procedure, now we're able to do again through a needle hole percutaneously. Talking about venous disease, a lot of patients that have deep venous thrombosis, they can cause a lot of pain, varicose veins, ulcerations. Here in the top left, this is a chronic occlusion of the patient's inferior vena cava. You see all these snake-like tortuous vessels here. These are actually perivertebral plexus. These veins you normally should not be seeing on a venogram, but the blood from the lower extremities has to find a way to get back to the heart somehow, so these collateral veins form. On the right here, this was after a complete reconstruction of a new vena cava for this patient. And you can see immediately at the end of the procedure, you don't see these spider tortuous collateral veins anymore. This patient now has nice, robust flow through this stent graft, this prosthetic material that's going to help with all of the venous symptoms and problems. Thromboembolic disease, Ravi had an example of a pulmonary embolus thrombectomy. So these are catheters that we can put into the pulmonary arteries to aspirate big chunks of clot out to relieve the patient's right heart stress, which in the acute setting can cause death, or in the long-term setting can cause right pulmonary hypertension, ventricular long-term right-sided heart failure, or chronic thromboembolic pulmonary hypertension. Okay. Peripheral arterial disease. So again, patients that get claudication or are having ulcerations of their toes that have arterial problems, we can go in with wires, catheters, use balloons, special stents. These images on the right here are atherectomy, actually taking away and trying to treat that atheromatous plaque that builds up, closing up the arteries there, okay? And so much more. So again, every disease process has an avenue where IR can intervene on in some way, shape, or form. There's various contrast agents, vascular closure devices, foreign body retrieval, intravascular imaging, a lot of different technologies that we apply to virtually any pathology. So now going back to Charlie, historically, a lot of interventional radiologists would be asked to do a procedure, you would do the procedure, and then you would be done. The future of interventional radiology, and we'll talk about the training path here real quick, is clinical interventional radiology is really needed for the field to progress. So now we're consulted to evaluate a patient. I evaluate that patient and figure out if there is a procedure or something that I can offer to the patient. I do the procedure, and then I also follow up the immediate post-operative care and see that patient in my clinic, okay? So what used to be more of you order a CT study, you do an IR procedure, now clinical IR is really the future of the field, and it's why the IR residency has in essence come out, okay? This is basically just summarizing what I just said. Historically, IR was a subspecialty of radiology, so you would do a diagnostic radiology residency followed by a 1-year IR fellowship. Now, because IR is so clinical, there is now a IR residency. It's a unique primary specialty. It's no longer a subspecialty of radiology. You are still board certified to do the diagnostic imaging, but it's a unique primary specialty, okay? And I'll talk real briefly if you have any questions. These are the 3 main pathways that you can become an interventional radiologist. 2 of them are 6 years long. 1 of them is 7 years long. So the 2 6-year pathways, one directly from medical school, you apply for the integrated IR residency. If you match into the integrated IR residency, then you'll do your internship year, followed by 5 total years of IR training, okay? There's no additional applications for that. For the independent IR residency, this is either a 6 or 7-year pathway where you do need to reapply for a second IR residency during your PGY3 or PGY4 year, okay? There's pluses and minuses to both. I'm going to leave it open to questions, or by all means, for extra information, the Society of Interventional Radiology has a phenomenal webpage, particularly for trainees, rfs.sirweb.org. This is my email address. If you have any questions, don't hesitate to reach out to me at any time.
All right. Thank you so much, Dr. Tabriz, for your talk. It's really amazing how you could capture kind of the breadth of IR in such a short amount of time, but really appreciate you sharing, all the different procedures that IR can do and how it's moving, or how it is really like a clinical specialty. So yeah, thank you so much. We'll save questions for the end, actually. We're going to have a panel. So everyone, please keep your questions, and then, we'll ask those later. I also quickly wanted to introduce in the chat, Dr. Vatican Cherry. He has been, he's been kind of providing a little bit of like a real-time annotation of what Dr. Tabriz was talking about. But Dr. Vatican Cherry, he's a vascular and interventional radiologist at Kaiser Permanente in LA. So thank you, Dr. Vatican Cherry, for joining us as well. So actually, we're going to get started with our next talk. And Robbie, do you want to introduce our next speaker?
Absolutely. So our next speaker is Dr. Lindsay Mccann. Thank you for joining us. So Dr. Mccann is an interventional radiologist at the Vancouver Hospital and associate professor of radiology at the University of British Columbia. He is the co-founder of Angiotech Pharmaceuticals, which has developed and licensed the paclitaxel-coated stent. He has also issued nearly 140 patents. So a great person to talk to in terms of entrepreneurship. So without further ado, Dr. Mccann.
Thanks. Good morning. Let's do this with trepidation here. Can you hear me, Robbie? And can you see my screen?
Yes.
Great. Okay. So that was an incredibly inspiring talk. I actually am thinking I want to go back and apply for an IR fellowship after listening to that talk. And it actually does reflect our day-to-day. There are just so many opportunities in what we do. These are, I have to tell you about conflicts, but hopefully none of these will actually play into what we do. We're going to talk about entrepreneurship and innovation, and obviously that's part of what interventional radiology is. It's the lifeblood of innovation. But innovation really depends on your perspective when you're thinking about it. For instance, something that transforms people's lives in Mexico is just a telephone-based healthcare advice and triage, where a million subscribers pay $5 a month, including for their families, through their phone bill, and have, for the first time, access to the most basic aspects of healthcare. Innovation in the way that we think about it and in the Western world has really changed in terms of medicine. A lot of the things that we're going to talk about are really the kind of things that you see where they're just mechanical solutions to late stage diseases. That was all of Dr. Daughter's approach to medicine, was mechanical solutions. And then really where I got started was biological solutions, because so many of Dr. Daughter's innovations in themselves actually caused a localized type of disease, if you like. And then so much excitement went into simplification of surgical procedures, essentially around visualization techniques. And that still continues today, but really where the action is today is the simplification of patient care through digital technologies. And we're going to come back to that, but that really is very much the theme of innovation today. So when you're thinking of innovation, I think there's a few things you need to basically consider. We're going to go through some more practical things, but everybody starts out and they want to win a Nobel Prize and they want to, transform the face of medicine. But the inconvenient truth is that is really reflected in this graph. And that is the fact that healthcare is growing much faster than anybody's economy. And that, in fact, no one can afford to deliver every type of innovation that comes along. And in particularly in the United States here, where the cost for life expectancy in terms of healthcare spending per capita to years of life is way out of line with everyone else. And in addition, medical errors are the 8th leading cause of death in the United States. And so really what I tell young innovators when you're thinking about where to go is follow the money. And so it doesn't mean that innovation isn't warranted. It means that the types of innovations that are likely to get accepted by those that are going to pay for it, which is something you always have to think about, should be and at the end of the day, be a more efficient way to deliver healthcare. And sadly, better ways are not necessarily going to be what's accepted. So here's an interesting idea. I think all of you are familiar with the idea of an EKG. Well, an EKG, really that standard QRS signal is about 5,000 bits of information per second. And most of it is just smoothed out. But in fact, there's a lot of information there, including vector. And so from what is essentially an EKG, this company called A4L has taken that information, and you can create a 3D map of a heart, and you can actually create a real-time beating heart, if you like, and calculate ejection fraction plus or minus 4%. And so what that means is instead of when a patient comes in and has chest pain, you can do this EKG. You can eliminate all of these other tests in the pathway and actually arrive at the same information and decide who needs a catheter angiogram or possible intervention versus who needs to be treated medically. And so it's a fantastic innovation, but the main reason it's probably going to get accepted is because, in fact, it saves a lot of money. And in addition, in medical systems where you don't have that infrastructure there with all of those other things, just like the way that cell phones leapfrogged landlines, this is a way to provide advanced cardiac assessment without having to have a massive medical infrastructure. So, you're all bright young minds and at early phase, and this sounds a bit cold, but the reality is, if you come up with an innovation and it's just a lab experiment, but doesn't actually make it into patients, it's just a lab experiment. It really has to make it into patients. And so reality is you have to follow the money if you're thinking about innovation. And so the present financial realities have decreased the relative investment in new devices. And really it's more about finding efficiencies or particularly quantum leaps in efficacy. And then finally, remember that what I told you about the 8th common cause being medical errors, safety. Safety is a massive area of opportunity for innovation in 2021. And so in 2016 already, money was not really going into devices. It was going towards telemedicine and digital devices, but there's been a massive sea change. In fact, between 2019 and 2025, in fact, and this is before the full impact of COVID and all the things that it's done to medicine have actually been observed. Is in fact, it was thought that probably about 3/4 of all medical innovation money was going to be spent on digital. In fact, it's probably not even going to be until 2025 because, of course, with COVID, we've had to have virtualization of healthcare being delivered. So we've gone from the digitalization to the Uberization of healthcare. And so there are massive shifts in the way healthcare is being delivered. It saves money, it's efficient, but it also deals with the realities of healthcare. And so the essence of healthcare change right now is convergence. And being engineers, this is an amazing time to come into minimally invasive therapy, in which interventional radiology is the leader. And so all of these things are being converged into medicine. You might say, gee, entertainment? Absolutely. Gamification of rehab services. So a couple of the leading stroke rehab companies now are basically doing all the things that you guys have grown up with and trying to get your grandpa who's recovering from a stroke now and doing this. And it turns out that you can actually, this is a great way to recover. And amongst all of this, the underlying it is data. And data, of course, something that Google and Facebook discovered and realized before the rest of us were paying attention, is that the data that's generated has enormous value as well. Value monetarily, but also value as a substrate for research. So you there's something else you have to think about when you're talking about innovation in medicine is medicine is not like any kind of normal marketplace. And so some of you may in fact have worked or done summer internships or yourself work at companies that develop consumer products, and you come up with a great idea and it actually solves a problem. Well, you get it into the market fairly quickly. Well, there's some actual things that are actually unique to medicine that are a little bit different, and unfortunately, you have to keep in mind when you're thinking about innovation. And some of them, in particularly, are funding. So, who's going to pay for it? So, you have these oligarchies in the United States that deliver healthcare. In Canada, we have basically one gigantic healthcare organization across Canada, which sort of controls things. In Europe, it's a combination of private and public partnerships, but basically funding of healthcare is not a natural process. You have a lot of regulations that you have to think about. And then finally, customers. And I think here is especially where there's an incredible opportunity, again, is that we have increasingly engaged consumers of healthcare. When you're thinking about, we're talking about entrepreneurship, that was what I was given. So when you're thinking about getting this out there, you need to understand some realities about startup outcomes. So we're taking, in North America, we're talking about all types of innovation here. So about 9% of innovations found after 2010, these are ones that actually got to the stage of being a startup company, actually turned into a company. Okay, that was before the massive explosion of innovation and entrepreneurship that every university has now is now encouraging virtually every postdoc to do. Now that number is about 1 to 2% instead of 9%. And just as a sort of an aside, to tell you what it's like, so this is just from one of 3 entrepreneurships at the university I'm at, the University of British Columbia, is generated. So last year we had 472 startup companies that actually became incorporated as companies. They raised $290 million and generated $34 million in revenue, which is a different story. But it's just a way to say that just one of 3, about 472 companies, there's a massive amount of innovation going out there. And what that means is you better run like hell, okay? So this is a common thing, innovation funnel, the idea that comes in, you're going through this process, and it doesn't mean don't do it. It means if you're going to do an innovation, it's not something you can kind of do part-time and think about. Once you come up with an idea, if it has value, you better run like hell, because someone has to be thinking about this all the time, because there's a tremendous amount of innovation and all of them competing at the end. So this is a really great talk. It's getting a bit old. It's a TED Talk from 2015. Bill Gross is one of the amazing innovators in the United States. He has founded and fostered an enormous number of companies. These 200 companies were all companies that he had some role in. He looked at 200 companies, and what he said, I think still really plays. And that is what matters most for startup success. And he found that the 5 factors, timing, team execution, whether the idea was really something that was really an outlier, something really out of the box solution, the business model, and then funding. And timing overwhelmingly is the most important success in launching a company. And so, and basically, for instance, his own company came with this idea for a video content business, but it was before you even had the internet, before you had broadband and the software available. So basically it was YouTube, but they launched it before the problem could be solved. So basically, YouTube came along just as the internet broadly became accepted. And so you've never heard of Idea Lab's version of YouTube, but you certainly have of YouTube. All of these other ideas are things that are important, but if you come out too early, the infrastructure that's needed to support you is not in place. If you come out too late, you've lost that competitive advantage. So Ravi asked me to talk about some mistakes I made. So I was in 1988. I was working in London, England, and at that time, very different place in the United States. Basically, if physicians felt that something was a worthwhile procedure, you could actually go and do it. You didn't have an overviewing FDA. Very different now. And this guy named Hans Wollston, there's a stent called the Wall stent, and people don't realize it's named after a Swedish gentleman named Hans Wollston, who was in his own way very much like Charles Dotter, very much an iconoclast who came up with his own ideas and really wasn't going to didn't care about pushing boundaries. By 1989, 1,200 of these had been inserted, and they weren't working very well. They were causing all kinds of problems. So here's a superficial femoral artery. That's the femur right there. And there's the wall stent in there. And I think you can see that there's narrowing within it. So putting this stent in to treat a little narrowing that was there beforehand, in fact, induced its own kind of reaction, if you like. So an overreaction to the reparative mechanism that was there for the stent. And I tried all kinds of things, radioactivity, heating, electrolysis. None of them worked. And I actually moved to the United States because I thought I was going to work with a guy there, and it didn't work out. And I actually gave up, and I thought, okay, that's just one of those ideas. And I came back to Vancouver, and I actually went to Thursday morning vascular surgical rounds, and one of the vascular surgeons who was supposed to be presenting had slept in, and he had forgotten, and he had a medical student working for him. And I said to the medical student, do you have anything to present? Because it's my turn and I don't have anything. So this guy presented cartilage-derived angiogenesis inhibitors in animal models of rheumatoid arthritis. This is a picture of the room, and this is a picture of all the people that were in the room when this guy started talking. And I was sitting in the back because I felt sorry for him. And this guy had exactly what I needed for a stent coating. And so I would say to a first message to you as a young entrepreneur, honor every opportunity to learn and keep an open mind. My mouth dropped. Here I had traveled literally around the world. I donated 3 years of my life trying to find something. And after giving up, this brilliant medical student who eventually became my partner in Angiotech, I started this. And basically the principle was that new blood vessel growth, which is called angiogenesis, it's a process where any tissue that grows, it has to actually develop its own capillary network, except in cartilage and in cornea. And so the idea was if you could actually control blood vessel growth, you could control tissue. So if you put these inhibitors on a stent, no tissue could grow more than 2 millimeters away. Pretty naive idea. It turns out we were wrong for the right for the wrong reasons. And cartilage, it turns out, didn't have enough biological activity, but we actually got it started. And we did some basic science where we took a fertilized chicken egg and we grew blood vessels on the surface. This is how normal blood vessels grow, and it's called arborization because it looks like a tree, and you can observe it in tumors. You can see how they cause new blood vessels to grow in. Anyway, we got some money from people who actually turned out to be mafiosi. I'm not kidding. And we way underestimated how much we needed. And it turned out our experiments weren't working, and I was really sweating because I was kind of worried about what these guys would do. And it turns out that the bark of a yew tree actually did exactly what we needed. It was really a bit of serendipity. We basically did some animal models, and then I got in an airplane and started traveling all over the place. And one thing that you'll find if you ever actually make a device and you decide you're going to interface with companies is that companies are very slow to make decisions, and all said they would be making a decision soon. And we actually only got a deal from the two people who put it out there after they thought a third company might be buying it. And so if you, it's like anything else in life, if you go to a dance and nobody looks so good as the person who's dancing with someone else. And that's true in life. It's called deal tension, and it's a way to get anybody to interface with you. And so another lesson that we learned is that the timetable will of large companies inevitably differ from your own, and all companies make decisions very slowly unless there's what we call deal tension. So we founded a company then. We went public in 1997. We did this co-exclusive with the two biggest companies. And another thing that happens is you get pushed aside because now you're playing with live ammo, and you really can't have people who are doing two jobs. And I'm an interventional radiologist, and I love what I do. I wake up every morning just loving what I do, by and large. And if you're not willing to give that up, you can't deal with a company that is worth a lot of money. Unfortunately, what happened was we learned some hard lessons, and that company eventually went bankrupt. Even though the reasons it went bankrupt, it turned out were actually fallacious. But at the end of the day, this thing has gone in over 9 million people. And between paclitaxel coated stents and balloons, and that's many more patients than I would see in my life. So you're struck by an idea. What should you do? Well, I tell people, even though invention disclosure sheet is kind of a dated concept, it's actually a really useful thing. So you can download these things. Just Google it, and you'll see. I just did this just before Christmas, and I got this many hits. There's a bazillion of these things online. But what it does do is it first of all starts talking about when you came up with the idea. That doesn't protect you, but it kind of creates a thought line. But it really is a great way to clarify your thinking, and it really makes you're going to have a lot of homework going forward, and this is a really great way to kind of organize your homework. This is what it does. It asks you some questions about what it does, how does it do it, why is it different, okay? And so I highly recommend that you go through this process if you do it. Then you need to think about how you're going to do this. And remember that what you need to develop is in medicine is a useful, approvable, and sellable product. It has to, what I talked about before, it has to solve a problem, preferably in a cost-effective or efficient way. It has to be approvable. It has to be get it through regulatory, and then somebody has to pay for it. There has to be a way to get this thing paid for. And these are the ways you can do it. You can fund and do the process yourself. You can start a company, which means you're going to convince others to invest, or you can license or work with an existing company. And when you think of all the ideas that it takes to get to a useful, approvable, and saleable product, even we're talking about digital products, you have to have the idea, intellectual property, make a prototype, try it out both in the lab and in the clinic. You need to raise money, license it, build, manufacture it, get regulatory approval, get reimbursement. Which of these did you cover in your training? I suspect, well, would probably be some smart aleck out there who tells me, yes, he's done his MBA and has approved 3 products, but most of us haven't. And so that expertise exists in spades in those big companies that are out there. And so for most of us, interfacing with big companies is actually going to be the best way, because another common error is failure to assign value to these skill sets. Every single step in that is absolutely critical. It's the weakest link in the chain. And in many cases, the skills required to do all of those things required every bit of training and hard work as what you bring to the thing. So I'll just finish with interfacing with large companies. Common mistakes made by entrepreneurs is the first one is a concern about getting screwed, which is, I think you all can do the math. Remember that discussions with a company do not constitute an agreement. And so a lot of people talk to a company and think that that's going to be success, and that's not true. Don't base your financial plan or exit on an aggressive timeline. As I told you about our own experience, it actually took years for us to get an agreement, even though ultimately the device turned to be transformational. And then what I really would tell you is that some great advice I was given is when you're struck by an idea, write an invention disclosure form. True medical innovation requires many non-medical skill sets and more time and money. Standing rule is double the time, double the money that most practicing physicians have, and the most practical route in most instances is interfacing with existing companies. And I think I'll stop there, Ravi, but I'm happy to answer any questions that may come up.
Yes, and thank you so much for the talk. That was very amazing to see what you've done and all the wise advice you have for innovators in the future. I think that we're going to save questions for the panel continuing, but thank you again. This was an incredible presentation. And now I will pass it on to Pratik to introduce our next speaker.
Thanks, Robbie. I'd like to introduce our next speaker, Dr. Arvind Arapalli. He's an interventional radiologist and medical device innovator at Piedmont Healthcare in Georgia and an adjunct professor of radiology at Vanderbilt. Along with his practice, he's an active researcher and holds multiple patents with his work in MRI, device development, and minimally invasive therapies. He's the co-founder of Surefire Medical, now Trisalis, a major medical device company focused on developing minimally invasive devices for the treatment of cancer. And he will talk about his own entrepreneurship journey. So please join me in welcoming Dr. Arapalli.
Good morning, everyone. Can everyone hear me?
Yes.
Okay, just start here. So I think this is a talk that I'll sort of follow Lindsay's talk. Lindsay did a great talk. It was a really good high-level perspective of innovation, medical device development, especially on a perspective. And one slide that really caught my attention was Lindsay's slide on the innovation funnel, where he says you got to run like hell. And I think this talk will sort of summarize that innovation funnel that I went through in terms of being able to bring a device from an idea into clinical practice. So I'll share this, my perspective. This is going to be more like a one perspective of how we did something and the process involved. If you look at any device development in general, there's actually a standardized process that's available that you can look at, that there's milestones or steps along the way. Here's a nice chart where you have the value of the company on the y-axis, you got the milestones on the x-axis, and you can see as you move from concept to prototype, patent applications, and clinical trials, the value of the company goes up. And that's sort of, for this talk, I'll sort of walk through this process we went through at each milestone and what it took to get there and what we learned along the way. To first start, I think one thing that really helped us give us guidance is the Stanford Biodesign model. This is a well-known, established process for medical device development. Started at the Stanford Biodesign model. And really, when we started our company, we didn't really have an idea, but what we did is we decided to go through the Stanford Biodesign approach and see if we can start a company based on this methodology. So using the Stanford Biodesign model, we really have three components. First is identify, which is identify the clinical need. And then from the clinical need, then you invent and then you implement. And that's really the approach that we took when we first started. Like every really startup, you really have, it's not one person, it's a team. And I started first with Jim Chomas. There's Jim here. He's a biomedical engineer. He and I met through a mutual friend. Initially, the way we started the approach was we went to looking at different clinical needs. Jim would travel around the country, go to different IR lab suites. I would have a notebook and I would basically write down clinical needs as we were working through this process. We did a needs identification. And some of the criteria we used when we were starting this process, we wanted to work in the medical device space. It's going to be somewhere in IR. We wanted to have a low-hanging fruit, and we want to do something with a more straightforward regulatory pathway, since this is our first time trying to do something along these lines. And this is sort of how it started. And we went through multiple clinical needs where Jim and I would talk about once a week, would meet via Skype or other methodologies and talk about the different clinical needs, and we'd slowly pare down. And I would write down 10 needs in a week or 10 needs in a day. You do the same thing, and then we'd sort of eventually break it down. And what we ended up doing was we finally fine-tuned our clinical needs into one specific area of interventional radiology, and that is the delivery. We've noticed that there was a lot of advancements in imaging technology, especially with some of the larger vendors like Siemens and Philips. And we also noticed that the therapeutics were really improving well, such as the use of chemoembolization or Y90. The agents were growing, but what we noticed was the delivery of the agents were the same. And so what we decided to focus on is, can we do a better delivery catheter, a catheter that can serve as a microvalve to help prevent reflux and also improve targeted therapy. And that was really the gist of how we first started utilizing our ideas. Well, like with every company, there's always a bar phase and a garage phase. This was the bar phase where Jim and I first met. This is a bar in Atlanta, in Piedmont Park, where Jim and I first met. Like I said, we met before we even started the company. We met first doing clinical needs. And then here at this bar, we sort of figure out on napkins how we want to approach the idea, the clinical needs. And it was like two kids in a treehouse. All right, I'm going to be the CEO. You're going to be the CSO. Let's sort of do this. But it started on a napkin. From there, we slowly built up. In the bar phase, what do we do? Well, this was actually, I would say, the funnest part, where we did extensive research. Jim and I went back and we did a deep dive into the clinical need problems. We did a deep dive not only into the published literature, we looked at the patent landscape, and we looked at the regulatory aspects, everything along that side. After that, then we wrote a business plan. We incorporated Surefire Medical, but then we wrote a business plan. And if there's anything that you can really help yourself, if you have an idea to see how well it's moving along, is to really do a business plan. And this is a copy of the original business plan that Jim and I wrote together. And to be honest, doing a business plan is like writing a grant. It forces you to think about all aspects of the company. It allows you to organize your thoughts better and also do a better analysis of the deep dive. After that, then we moved on to what we call the garage phase, where this is where we really formed the core team. We've tried to identify engineering help. We tried to identify IP attorneys, and then we found regulatory experts. Now, again, at this stage, we're really bootstrapping all of our company. Jim and I both put money into it ourselves. There's Jim, and actually, this is a picture of our first engineer we work in his garage, and I kid you not, his name is Ron French, last name French, perfect for a catheter design engineer. But that was our first engineer, and that's how we first started doing all that. So we went from the concept, and then we'll go through the other things. But one of the things I learned is that there is a aspect of fundraising that sort of is important for everyone to understand when you're doing these kind of medical device development. And this is sort of what I call the startup calculus. First, you're going to have angel round or your own funding, which is basically you're having people provide, friends and family yourself provide money, and that will sort of get you to a certain stage. And here in our situation, we went to a basic early prototype, did our patent application and everything else. But along that way, you're going to burn through that money pretty fast, and you're going to need some more money. And this is where we were, where we had done some early feasibilities and patent application, but we really ran out of money early after bootstrapping. Next, you got to then raise more money. And this is what we call Series A. And where with the Series A, you're going to be using that money to really drive theoretically if everything works well, going from animal testing, human clinical trials, and down to FDA approval. And that's the burn. That's basically you use that money to establish that. And then the next step, what I would say is potentially Series B, is you raise more money. And theoretically, with the Series B money, you're then able to do a market introduction, and then eventually you're able to make money, and therefore you have a sustainable company. And that's what I would say is the general concept of most startups in medical device. Obviously, Series A and B could be different, depends on how far along you're on the process as you're raising the money. And then the burn rate is basically how fast you go through the money from the angel round, Series A and Series B. And these are all important when you're talking about aspects of the money. The other thing I want to really emphasize, and which Lindsay also had mentioned, is that everything is going to cost double what you think in terms of time and money. And to be honest, a lot of my mentors have told me time is your biggest enemy. You need a lot of time if you're going to do this. This is not something you can just do, a few one day a week kind of thing. You really have to invest a lot of time to do this. It's like building something from zero. It takes a lot of energy and effort. It's a lot of fun, but you have to understand the time component of it. Here's another aspect of when you're raising money, how it works. You have the seed money, which is about 50,000 to 1 million, and Series A are different amounts of 1 to 5 million and 5 to 15 million, or Series C is 10 to 25 million. In general, this is the amount of money that's raised, and you can see the dilutional aspect that happens. So as you're raising money, as you further go along the process, just remember there is a dilutional component of it. You can do some mechanisms where it's not diluted. In other words, you can do some SBIR grants that's non-dilutive, these kind of things. But remember, there is the check of reality is as you're raising more money to fund your project, you're diluting more of your ownership in general. And these are some of the hierarchies of raising money in general. If you want to start talking about, if you have an idea, you want to do it, you can do it yourself. You can do SBIR grants, SDTR grants. I know a lot of my friends that have done SBIR grants, and it actually works really well. Again, you can raise not a significant amount of money, but you can raise non-dilutive money. But the aspect is there is the downside to that is it's an NIH grant application mechanism. That's one. The second thing is there is a time factor in that government grants don't obviously happen quickly. It's possibly a 1-year process commitment to going through the process and finding out if you do get it. And then also you may not get it if you go through the process. So just realize that. Then you have the family and friends option. Then you have the private placement memos, and then you can then move to the parts of angel funds, venture capital, and then eventually IPOs. And these are, I just want to give you an idea of where the hierarchy of raising money. For Jim and I, when we first started with ourselves. We did submit some SBIR grants, which we did not get, but then we moved quickly to angel funds. And then mostly we went to some angel funds and we ended up getting some venture capital money. Now, if you're going to go to venture capitalists, I think you sort of have to know some general outlines and rules about as you move along this process. What did the venture capitalists look for? They're not looking for another Me Too product. They really want an elegant solution to a major problem. And I think some of the highlights that Lindsay had shown in his talk is a good summary of venture capitalists and where they're going right now. Market potential of really over 500 million because when they want to invest, they really want to look for a high return on the investment. They look at the product fails, what the team will come up with. So what is the core of the team? And that was at the beginning, since Jim and I were new entrepreneurs, really to bring people onto the team that had some experience. And that was very helpful when we were going for fundraising. And then the venture capitalists want to know is how are they going to exit their strategy? How will they get their money after all this? So that's sort of a concept that everyone has to look at when they're going to go for a venture capitalist. And then they're going to look at past successes. What was your success rate? Who are the people involved? And are you a known entity in general? Obviously, the longer the track record you have, the easier it is to raise venture capital money. So then here's the next step. What we did the application, we raised Series A, then we moved to animal testing, and we actually ended up doing first in man clinical trials. This is an example of our first prototype. This is a microcatheter with a valve on the end of it. You can see, and this is literally my kitchen sink. I took a picture of it. This is the initial animal testing we did, where basically we went and there was a private animal facility here we used. You can see the device, you can barely see it. We put this in this catheter. Our first initial prototype was a 9 French catheter. You can see how big it is in comparison to the renal artery. And you can see early prototypes showing that we prevented non-target injury and no reflux in this. And so that shows you how we went from a 9 French system eventually to now where the system is, the microcatheter itself was about 2.5 French and will go through a 5 French catheter. But as you can see, the prototype is going to be crude, but at the same time, these are prototypes and you have to improve the modification as you're going along. That being said, we did a first in man in May 2011. So we actually went very rapidly, starting from 2010 to 2011, where I actually went to have a product that was first in man capability. I went on a 4-day trip to Sydney, Australia, to do a first in man trial. This was a mechanism where Australia has a process where if there's a Australian company that's willing to sponsor you, we can do a first-in-man trial. So they offered us in January, and we had 5 months to get the product ready. We were able to do this, so I flew to Australia. This is the Certex team. We flew to Australia. You can see Lawrence Bester, David Cade, or Lawrence Bester is an interventional radiologist in Sydney, Australia. David Cade was Chief Medical Officer of Certex in Australia. And then this is Dave Liu, who is in Vancouver, interventional radiologist. They all flew down. This is me, and we actually did the first-in-man over here. Ironically, I flew all around the world to do the first-in-man and to find out that we could not see the catheter under conventional X-ray fluoroscopy. So it was quite the learning experience, but otherwise it went well. We had to actually end up going back and putting markers on the catheter so it could be seen under X-ray fluoroscopy. But that's basically how we went through the process. So that's our journey, how we went. I won't belabor all the other details, but I just want to give you highlights of how we went. So what about some tips and tricks of people that are interested in this? What can you do as you're going forward? I think the most important thing is you need to find an ecosystem. You need to have an environment, mentors, and team. This is not a solo effort. Whenever you're doing something like this, you need to be surrounded by people. You need to be surrounded by mentors, especially. And so that was important if you're going to be doing any kind of innovation. You can't be doing this by yourself. You really need to be with people in a team. That's one. And where can you find some of these ecosystems? The good thing is there are all kinds of entities available. On the left, on the white background, you see some academic centers that have these kind of ecosystems. The Stanford Biodesign, Northwestern has what's called NUCAS, and then Hopkins, which I was involved with when I was at Hopkins, called CBID, Center for Bioengineering Innovation Design, which are basically biomedical engineers teaming up with clinical faculty to try to work on different kinds of clinical problems. On the private side, there are actually, I just list some in Atlanta, where I am. There's the GCMI, there's Hatch Medical, and then which is a incubator, and then there's a pediatric consortium for device development. So wherever you are, you can sort of find a local, geographically, a region that's geographically friendly for you to find some of these ecosystems. And I would encourage you to find some of these, go to the meetings, start networking and trying to find people that are available in this area. The second thing is, I think innovation is the ability to take a risk. I think you have to be able to be willing to step outside a little bit your comfort zone. I had to learn, I was comfortable in my prior job at faculty at Hopkins, but for me to have to step outside to learn a little bit on the business side, also regulatory side, intellectual property. It's very important to sort of stretch yourself and be comfortable in new environments. And this is an example of, you have to realize this is from a venture company I found slide, but basically what happens is you got the pain and pleasure scale on the y-axis, and then you got the years of funding. And I'm sure Lindsay can probably attest to this, but you're going to go through this cycle of pain and pleasure where initially you're going to be at a high, and then you're going to go through some very difficult times when there's no funding, there's everything's falling apart, it seems like, or there's all kinds of other issues. So just remember that innovation takes a risk, and it's not all fun and games. There's a lot of difficulties along the way. Other things I think important thing I want to suggest for people is to challenge yourself to conventional dogma. And I show this slide of the Wright brothers, and they're a classic example of challenging the dogma in the sense that when if anyone reads about the Wright brothers or the book about the Wright brothers, was just fantastic. But basically, the dogma at the time was that to fly, you need really powerful engines. And that was how you need to, how airplanes were going to be built. And that's what everyone was. And if you look at the Wright brothers, they were basically bike mechanics. And they completely challenged the dogma of thinking, because they said, maybe it's not necessarily the power you need, but maybe how you can warp the wings to control flights like birds. Birds don't have any significant power, but what they do is they capture the wind, and they're able to sustain flight by warping it. And that was the whole concept of the airplanes. And having warp wings allowed them to fluctuate and control flight. And so that's a classic example of challenging dogma. There's so many dogmas right now in our culture of medicine. And so be willing to challenge the dogma when you're going through innovation. Finally, I think read. If you want to be going through innovation, it's just going to be a process of also reading. Here are 4 books I recommend, which really have taught me a lot. First is the Biodesign book with Paul Yock at Stanford. It's a great textbook. I use that as a reference. A book that I like called Venture Deals, Be Smarter Than Your Lawyer, Venture Capital is really good for me, a good book that sort of gave me an idea on the aspect of the finances of venture deals and what you can learn from that. There's a magazine called Startup Magazine. It's got really good ideas, and they basically highlight different startups coming up. If you're interested, you can sign up for that. And then there's also a Entrepreneur's Guide to Biotech Startup by Peter Kalinsky, which is also a good source of knowledge for all this. So in essence, I think just be creative. It's a lot of fun being doing some of this. Some of the most exciting and memorable fun times I've had was going through that startup process. It's a lot of fun. It pushed my creativity, it pushed my knowledge base, and really, to me, it was also extremely satisfying because the people you meet and also the impact you have. So I finished with this quote: "The reasonable man adapts himself to the world. The unreasonable one persists in trying to adapt the world to himself. Therefore, all progress depends on the unreasonable man." So I want everyone to use this quote and be unreasonable in their thinking. And these are all the people I want to thank along the way, with my wife and daughter who were supportive as I went through this. And then, of course, all my friends and along the way that have helped me. Thank you.
Awesome. Thank you again, Dr. Arapalli. That was an amazing talk. Yeah, I think both talks really highlighted like a very high level and then a very specific walkthrough to help you visualize the process. I think that's helpful for everyone. So I think we have time for one question, if anyone wants to unmute and ask it. All right. Oh,
I'm going to ask a question. So, Dr. Arapalli, what do you think, in terms of young entrepreneurs, maybe can you just talk about the difference between wanting to make money and wanting to solve a problem? And how important do you think that is?
Yeah, I think, thanks, Lindsay. That is, people, I think, get blindsided by the money aspect of it. And as the biodesign model show you, clinical needs are so important. And that's why we're doing all this in the end, to make an impact on patients and to make an impact, a broader impact than just one patient at a time. So I think it's important to sort of focus on the clinical needs more than anything else. And, excellence in that, and excellence will provide success, and don't chase monetary reasons. It's easy for thinking that, oh, I have an idea, I'll make money. And if all your decisions are made based on that, then it's just going to be basically a blind ending road that goes nowhere. Whereas if you pursue clinical excellence and pursuing the right idea, then success will follow no matter what you do.
I would say that talk that you just heard from Dr. Arapalli is one of the best talks I've heard about innovation. You guys should record that, and people should learn. That was fantastic. And what he just said, I think, is almost the core of medical innovation. And can't stress enough how wise those words were.
Well, thank you to you both. If anyone has any further questions, feel free to direct it to our next panel session. So next up, we have our final event for the meeting. So we're going to hear from our expert panel of interventional radiologists, and they're going to be answering your pre-submitted questions. So Just to introduce them quickly, we have Dr. Rakesh Navaluri from the University of Chicago Medical Center, Dr. Cliff Weiss from Johns Hopkins Hospital, and Dr. Agnes Solberg from CHI St. Alexius Health in Bismarck. So we'll start off with some of the pre-submitted questions, and if you are so compelled and have some more questions from the talk, please feel free to put them in the chat or message me privately, and I'll see if I can get to them during the course of the panel. So just to start off, could our panelists introduce themselves and tell us why they were interested in interventional radiology? And we'll start off with Dr. Weiss.
So the reason I'm interested in interventional radiology is sitting 4 screens away from me in a yellow room with a blackboard next to him. So you just heard a talk from Dr. Arvind Arapalli, and it was my 2nd year of medical school. I won't even tell you when it was for both Arvind's sake and mine. And I after surgery, very soon in my 2nd year, which is when we start our rotations, I wound up very pointedly in interventional radiology, and spent about 6 weeks, I think, in old room 3, an old round II, putting in lines while Arvind and Mike D'Ignazio learned how to use their Palm Pilots, and was given a lot of leeway to kind of do fun things with these guys. And what I realized up front right then and there was that this was the answer to everything I was looking for kind of in medicine, right? It was diverse, that they were doing procedures all over the body. They were doing new innovative things every single day. They were being creative in their procedures. Even in basic procedures, sometimes you have to get creative. And yet they still had clinic, and they still had, they were still someone's doctor, even though there was a big argument at the time about whether or not that was the case. That people knew us as doctors, it definitely was coming. And I got to see really interesting innovation, even right then from the IR team, whether it was interventional MRI or at that time, liver cancer therapies were just getting started. And to me, I was looking at the future of medicine in progress. I think the other thing, as a research wonk, that really turned me on to interventional radiology was we had way more unanswered questions than we had answered questions. And we had a long way to go. And I thought, not only can I make a significant mark, but I could actually help the field move forward. And so that's sort of when it clicked in, and it didn't take much reinforcement for me to figure that out. In fact, I will argue that diagnostic radiology was a slog for me. I remember looking at the board. I would sit there for the day. Back when I did it, we didn't have these newfangled mice and computers. We actually looked at a rotating board. I'd get sitting there on my board, and I'd start working, and it was like one of those terrible, TV scenes where I worked and looked up at the clock, and 10 minutes had gone by. And I would spend an hour in interventional radiology with great people, and I barely work, and I'd look at the room, and it was 3 o'clock, and day was winding down. So to me, it just fits.
Navaluri?
Yeah, I don't know if I can have a more eloquent answer than Dr. Weiss just gave, but when I went to medical school, I was very procedurally inclined, and I really wanted to do surgery, and so I always I was investigating cardiothoracic surgery, maybe surgical oncology, and I was scrubbing into all these pretty complex cases. And one of the things that always came up was, these surgeons would inevitably, in a lot of cases, go to IR, and I would go with them. And whether it's something as sort of routine now as a TIPS, or coming up with some other alternative to a surgical problem that they couldn't handle, I mean, it really became apparent to me that the interventional radiologists were really the kind of the go-to for a lot of the physicians, especially like a lot of the advanced surgeons in the hospital. And that kind of piqued my interest. I was like, I want to be one of those people. I want to be someone who other surgeons can come to when they're in trouble and they need help, solving an issue with the patient. And so I think that was my initial kind of exposure to IR, was was seeing how much they impacted or helped out, their surgical colleagues. And then over time, what I really appreciated is just, the minimally invasive nature of things, and seeing the things that we're able to accomplish with just, essentially a needle, catheter, wire, is really, really amazing. Some of the things I've been, fortunate to be able to start doing in my own practice, and I've seen other people develop, has just been truly amazing. So, again, the term MacGyver is thrown about a lot, but, being able to MacGyver new ways to do things is really something that brings me a lot of joy. And every day I love having that challenge being presented to me.
Awesome, thank you. And Dr. Solberg just joined our call. Hello.
Hi, I am
Hi.
so sorry, guys.
No
I'm in the
problem
back
at all.
seat.
No problem. So we were just
So
doing
sorry.
introductions. So could you tell us a little bit about who you are and why you're interested in IR?
Sure. So my name is Agnes Solberg, and I am an interventional radiologist. And I used to be an internist. Actually, I used to be a hospitalist, and I just didn't like that very much. I was always interested in a more of a surgical approach, I guess, to thinking. And I kind of like the instant satisfaction of caring for patients and having a direct, having something happen right, having a nice result right away. It's just my personality. And so I really, that's one of the main reasons I like IR. And I also like toys. So I like to have all the different products and try to test things out. I really like to use technology with, in my work, and, IR is a rapidly advancing field, so things are changing all the time. We try new things. We pull stents off the market. We get new stents in, et cetera, et cetera. So it's very exciting.
Great. And just to get into some of the questions that, we got a lot of in the pre-registration phase, if we could go around and maybe talk a little bit about how does your day-to-day role as a physician relate to developing or innovating new medical technologies and techniques, and how do you interact with those things on your daily practice? We can start with Dr. Solberg.
Sure. Yeah. So for me, essentially, it's always fun to think of new things that we might need in our practice and be innovative. Just the other day I was doing a radio procedure with like the SIR catheter, for example. And I said, I really want a softer catheter to get into the little bit further for Y90 treatment into the hepatic artery, common hepatic artery, maybe even proper hepatic artery, if not even the right. So then, I asked the rep, I was like, hey, is there anything else that's longer and softer? And they've already developed it, which is kind of nice, but it's kind of exciting to always be thinking about new things, or asking companies to develop new things. Like, for example, with the BioPins device, I always give the reps and their bosses feedback on devices, because that's how you get things that you need, is by giving them feedback, and then they'll make what you want, right? So like the side button on that, I'm sure I wasn't the only person who wanted a side button on the, on the biopsy device, but now there's a side button and a better safety, et cetera, et cetera. So those sorts of things. And, I talk with some of my IR friends too, and make sure to see if there's anything that already exists. And if not, I'm not actually personally involved in the development of anything, but I always do have lots of ideas. And so I'm always excited to talk about with folks about things that are developing in the future.
Sure, go ahead, Dr. Weiss, Dr. Navalori.
Sure, I'll jump in. So I think that I don't have a biomedical engineering or any kind of engineering background myself, to be honest. I think my interaction with new technologies is really sometimes being at that big institution, we were approached by companies, and they may ask us to participate in a phase 3 study, sort of validate a new product, prior to getting FDA approval. Or maybe even more commonly, it's just new device comes out and we start using it. We have unique opportunities maybe to use it off label, and we end up publishing our results with it. So, our experiences is sort of, we try to have a pulse on what's being developed and try to be, I love trying new things out, especially if I have a unique patient or situation where it can be helpful. Of course, all whatever we do publish or our interactions with, device manufacturers, we do give our feedback and sort of as Agnes mentioned, we have some input. And so that's kind of how my role is. But again, I do want to caution them. I'm by no means a developer or engineer myself.
So I have a slightly different approach and experience by nature of sort of what I do. I'm involved with device design at kind of all levels. I'm actually quite terrible at the last stage, which is moving it out into the world, which is the nature of being in the same academic institution since I was a fetus. I'm very involved with the CBID team. I'm the medical director. I have been for a while. And so I'm always trying to shuttle ideas to the engineering school on a daily basis to get things designed, to have them vetted, to see what's real and what's mystical. And I try to meet a couple of times a month with some of the more intelligent and creative people in Whiting School of Engineering to think about, not only do I need to bring them ideas, but if I know what toys are coming down the pike, I can think about how we can take those things and apply them to what we do. So I try to meet at least once a month with our material science team or with our big data team and think about how we can apply what I never heard of before to things they've never heard of before, and to actually actively seek out new life and new civilizations, so to speak. The other thing is that I really do enjoy when companies bring me products early, often before they've gone through final approval, or maybe they have some stage of product that has come through approval and other that hasn't. I get a chance to bench it. I get a chance to test it often in an animal lab and give them feedback on whether their product is something we want to try or not. And then, of course, when new product comes, we all like to try it. I particularly am the go-to guy for a lot of the embolic devices at this point, what embolization devices and some other devices. So I really am involved at many levels. I've done a few attempts at startups. I think what Dr. Arapalli said earlier is very true. It requires an intense commitment of time, which is something that I'm lacking at this point in general. So that's why a lot of my companies are spinning out to students like, who are students who can then run with it and use their energy and their non-risk averse behavior to go ahead and try these things. So I really like the process at all levels. I think even just trying and failing, I learn a lot about what will work in the future. I'm always poking holes at things of what are we doing? Why are we doing it? If someone says, oh, I could never do that, that's a great answer to why we should be designing something, right? If someone looks at the procedure and says, how did you do that? Well, the answer is you should be able to do it too. And so let's make it so you can. So, different because it's actually part of my day-to-day life to teach this and to kind of work through it. But, I see more than any other specialty group in the world, IRs are always doing this. Every single IR is always thinking about how do you make it better, or at least they should be. And how do we figure out the next step?
And on that note, ties nicely to the next question. What do you see are the big areas of innovation in interventional radiology? You can talk about that within different specialty areas. I'll kick it back to you, Dr. Weiss, for this one.
I'm going to keep this one brief, which is not usual for me. It's a huge question, right? So I will tell you in general terms, we are not doing today what we did in the early 90s and late 90s, right? When I did my first rotation, there were still multiple, at least in even the academic practice, cerebral carotid angiograms, peripheral angioplasty, just started some oncology work. Venous was not really taking, we do new things every single decade, right? So prostate didn't really exist. IO wasn't a mainstay of what we do. Fibroids were taking off but they hadn't really kind of hit their peak yet. Right now we have geniculate coming, prostate coming, and I would challenge everyone out there as these new procedures come out to, on a research question, to ask, you know, which we're doing really well right now, which is what are their outcomes, but also ask the question of what are we actually doing? What's the mechanism, what we're doing, right? That's why. So I'm just going to not talk about devices, et cetera, but I'm going to talk about how the things are changing always. I think we're going to be looking at more targeted therapy, molecular-based therapies. We're going to be looking at therapies that put us out of business because they're given by mouth or they're given by vein and they are targeting a molecular mutation. And I think for interventional, it's going to be, you know, moving more towards truly personalized medicine, whether it's inducing an immune response or whether it's delivering a new agent or whether it's even doing high-level biopsy to actually see whether or not we're doing what we think we're doing. So I'll leave it to everyone else, but I just think it's almost impossible to ask that question because we don't do anything now in the same way we did it in 1995.
Yeah, I guess I'll jump in here. You know, I think that really IR can, you know, have an impact on any disease process, really. That's one of the beauties of our specialty is that we can have a hand in improving care at every level. You know, I think that, you know, currently I would maybe argue that cancer is kind of the biggest area of growth, and certainly there's been a huge, you know, increase in the last decade. But, you know, if you just kind of go through, you know, the top 10 causes of, you know, morbidity or mortality in the United States, I mean, every single thing, you know, really is something that IR can be involved in, whether it's cancer, stroke therapy, if it's trauma, you know, diabetes, for instance. You know, I know that, you know, islet cell transfusion is something that's, you know, hasn't really, you know, progressed as fast as maybe we would like it, but, you know, something that we do at our institution, for example, and maybe it's something that we can improve going forward. You know, kidney failure, you know, certainly in recent years, we've developed, you know, endo-AVF procedures for access. But there's so many, every single disease process you can think of, IR has a chance to have an impact. And so I don't think there's really one area where I would say anyone should focus on, you know, as students or as medical students or even residents. But just, you know, think of a disease process maybe you've interacted with, or you dealt with and you've had patients that have suffered from, and think outside the box. And, you know, I'm sure there's a way that IR can impact it that we haven't even thought of yet.
Dr. Silver, you want to add anything to that?
I'm sorry, can you say again?
Oh, would you like to add anything to that?
Oh, me?
Yes, the big areas of innovation in IR.
Sure. I definitely agree with oncology. You know, I think there is definitely areas where we can improve, not just with you know, like radioembolization and things like that, but with bones, cement, and, you know, some kind of devices to keep the cement in the bone, for example, or things like that. But like people are using stents, but there's really not a mesh that we can deploy percutaneously and things like that. But a lot of the problem is that, you know, some of these procedures aren't getting paid right now by insurers, and so it's very difficult to like for me, for example, as an interventional radiologist, to go to approach a company and say, well, I need this, it's not paid. And then, you know, how do I go to my society and say, hey, we need to advocate to get this paid? You know, I don't know how to do those things, like that part of it, because it's not what I do. I just help patients in and out of my day, you know? And so it's a little bit of a challenge for me when, like, you know, Dr. Vatican Cherry, like, he knows who I am. He's like, I am always full of ideas, but like for me personally, I always find this roadblock in like, well, how do we get over these steps? Like these, you know, like the reimbursement with CMS, and then again, like approaching companies and things like that. I just have very little experience with that personally, but again, I think I absolutely agree with oncology, and definitely vascular procedures, because, you know, vascular surgery is becoming endovascular more and more. So.
And the next question we got was, how can medical students and undergrads who may or may not have formal engineering experience get involved with interventional radiology? What are the opportunities that they have, both in the innovation side and then to also learn more about the specialty? We can start with Dr. Navaluri.
Sure. It's a good question. And, I want to fully admit that I don't have an engineering background myself, but what I will say is that there are a lot of opportunities, especially like this, this meeting right now that SIR and the RFS section are kind of putting together, where, you can participate in biodesign interest groups. I know we have a biodesign competition within SIR. And I think even if you don't have necessarily an engineering background or maybe experience, at least participating as a team member, seeing the process that goes into it. I know Dr. Arapalli mentioned the Stanford biodesign process, and so there is sort of a an algorithm in some ways to learn. There's certainly textbooks that have been written on those topics, and you can certainly read about that. But I certainly think that participating in events like this and learning, is just the best way to go about things, even if you don't have an engineering background. Maybe I'll pass it to Cliff or someone else.
So, I guess the answer would vary depending where you're coming from and what you have available to you at your institution. There are formal training programs. I love formal training programs. I would stay in school my whole life. I still sort of am in school. There are many biodesign programs out there, many of which are desperate to get physicians in, right? We in CBID always try to seed each of our teams with a physician if we can. And that can be a physician in training, that can be a physician proper, that can be someone who's been out in practice, because it adds a real nice perspective to that team, and does bring something. So many institutions already have things built in. There's a big biomedical engineering effort. There are physicians looking to build things, but I'm sure a lot of you come from smaller places that may not have that, right? And that's where you need to reach out to either formal education or informal education like this. But I think you've heard, from a lot of different people today and people on this panel with different engineering experience. I'm not an engineer either. That Yoda was right back in the day. It is do or do not, there is no try. Try it. Go find, if you have an interesting idea, you're probably a smart person who's creative and can watch a procedure, watch an environment and say, there's a flaw here, right? Why does it take Dr. Weiss 42 passes to put a needle into a non-dilated biliary tree? That's ridiculous in 2021, right? Or maybe it's not. Think it through, figure out what the problems are, talk it through with the physicians that are practicing. Now that's the answer of how do you get involved in biomedical engineering, again, of which Arvind's not an engineer, none of us are here, right? And yet we're still actively involved in it at this panel today. And I think if you start to build that interest, you'll start to find people around you who want to do it with you. We interviewed someone from CBID right now who was in medical school and thought up an idea, found an engineer, built it, sold it, and then moved on to the next idea. And that gentleman had never picked up a calculator probably since high school, knew nothing about engineering and design. So I think the answer is try, get involved. The same way if you want to get involved in research, just go be there and try and realize failing is totally fine. We all fail all the time and it's completely reasonable to fail, and don't be afraid of it. So, there was a question, an ancillary question, that was how do you get involved in interventional radiology, right? Was that or were you talking more about device design, Pratik?
Open-ended. Yeah, you could talk about IR specifically.
There are a lot of programs that the SIR and other institutions have to get you into IR, the summer fellowship, et cetera. If your program isn't heavy in IR, there are away rotations. But again, if you're interested and excited, most programs will let you in for a period to experience it.
Great. Dr. Solberg, any advice?
Sure. So the best advice I got in fellowship, in IR fellowship, when I was asking with, I rotated with Dr. Mahmoud Razavi at St. Joe's. I asked him, I was like, you know, how did you start a company? Like, how did you develop a catheter and this and that? And he said, he's like, how do you get invited to all these talks? You know, it's all this research, etc. You get asked for all these things. And he says, oh, it's, a lot of it is obviously he's good at what he does, right? And like, I mean, but he's like, it's all about connections and networking, Agnes. You have to get good at networking. And I said, well, I'm not good at networking. He said, well, get good at it. And I was like, oh, that's rough advice, but it was good advice because I did get better at it. You know, I worked at that part of it. So, and network where it's going to network in the areas of your interest, you know, and it's okay to reach out to people who you feel are above you, or, you know, in education or whatever. It's not a big deal. The worst that person can say is, no, I won't meet you. You know, now when I go to conferences, for example, I pick out, usually I look at the attendees or the presenters, and I pick out one or two people and I ask if they'll meet me for coffee, just to say hi and meet them, etc. The worst they can say is no, but like from the people who've asked me to meet, I've never told anybody no, unless I really didn't have the time to meet with somebody. And I've never had anybody turn me down so far. So, you know, I mean, the last time I was at SIR, you know, I had lunch with Dr. Geraldine McGinty because I asked, you know, I just asked. So like, you know, you just ask. And that's the easiest way to network, and network in the area of innovation and interventional radiology.
Awesome. Thank you. Thank you to all our panelists. Really enlightening. I think you answered a lot of the questions that people had in the chat. If you have a very specific like pathway or training related questions, you can direct them to Dr. Tabriz, who gave the first lecture today. Dr. Tabriz, you want to put your email in the chat or something if anyone wants to contact you?
Sure, no problem. I'll put that right now.
Awesome. Well, thank you. This concludes our panelist talk. If you could all stay on just for a few more minutes, we have some closing remarks and information about ways to get involved with future projects and learn more about IR that way, and work on some innovation opportunities as well. So I'll pass it off to Robbie for that.
Absolutely. Thank you, Pratik. And thank you to also, again, to all of our speakers for agreeing to participate today. It was amazing to get all your perspectives and a lot of very interesting presentations. So. Can everybody see this? Yep. Okay. So now we're at the part where we're going to start selling SIR a bit to all of our wonderful participants here today. So for medical students and the undergraduate students that are present here, SIR is a great organization to be able to get involved with. There's free membership as a student, so no cost associated, very easy to join. For the medical students that are here today, the Medical Student Reserves are a fantastic organization to be able to get involved with for projects. If you go on the SIR website that was linked earlier and just look for reserves, there's a very short application, and the application process only takes a couple weeks to join. There's a lot of interesting projects to join. This, conference itself was planned through the reserves. So there's a lot of really amazing chances to, meet faculty from around the country and other medical students, and, have really interesting events like these to kind of broaden your perspective on interventional radiology and innovation and a lot of other interesting things. And for the undergrad students, which is what this event was more or less tailored to, we're kind of starting a new initiative within the reserves for pre-med students called the Pre-Med Coalition. So this is a fairly new project. So for any undergraduate students, it's an awesome way to get involved, develop your interest in a field while you're applying to medical school, meet medical students from throughout the country and faculty, get involved in research. So we have your emails. We'll be in contact with you for future opportunities. And it'll be a really interesting way as an undergraduate student to get an interesting perspective into a new specialty and just continue that growth. So for research opportunities, this is once again for both medical students and undergraduate students. The biodesign competition is kind of the flagship project of our biodesign committee here in SIR. Undergrad involvement is encouraged, and I really want to bold that for all of our undergraduate students beyond joining the reserves. Joining these projects, kind of like what all of our speakers were alluding to earlier, your perspective is absolutely valued. As an engineer, as an undergraduate student, all the different perspectives make the group better. So that, joining those kinds of projects are a great way to get involved in engineering from the get-go, get involved in these innovative projects, and once again, meet people, network, and make connections that will be very helpful for your future medical school career. We're also potentially starting a new reverse engineering guide that I believe that Ravi has an example of that will be posted at some point in the future. So it's basically just taking currently used biomedical devices and describing how it functions, kind of educational, slideshows for the remainder of our members of SIR. Once again, a great way to kind of showcase our, engineering capabilities and how everything intertwines. And finally, we're introducing some mentorship opportunities. So why join SIR once again? SIR Connect is a very big program within the Society of Interventional Radiology, site. It's a great way to match with mentors. There's an open forum discussion that's very active with professors from throughout the country. People of all levels, you have attendings, residents, medical students, undergraduate students. It's an awesome way to be able to connect with people, online. And you could search your specific areas of interest. So you could really dive into the areas that you're particularly interested in and find other people throughout the country that are also interested in those areas. And we'll probably be happy to help you, develop those and give you projects. And beyond that, yes, the Interventional Radiology Interest Group. I just want to replug that the Pre-Med Coalition is a group that's just starting. So for all of our undergraduate students that are here today, it's a really, really wonderful opportunity to get involved with the project from the get-go and really be able to start something from the ground level and hopefully grow it and increase the amount of undergrad students that we're able to interface with. And I think that is the final slide. So I will leave this to Ravi for our final closing remarks.
Yeah, so again, thank you so much to all of our speakers for joining us for this event and all of our participants for, staying through. Again, we're really excited about, the different opportunities there are for biomedical engineers and people from all backgrounds really to join us in interventional radiology and, innovate. So again, I know there's a lot of different content that we covered today. We talked about innovation, entrepreneurship, and different ways to get involved, regardless of your level of training. Please keep an eye out for your emails. We're going to be emailing everyone who registered with different opportunities that there are to get involved with SIR and different engineering opportunities as well. I just wanted to plug the SIR Twitter account, so at SIR Specialists. It's a great way to keep up with the information, the most up-to-date things that SIR is doing and future events that we're going to host. And also, feel free to contact either myself, Pratik, or Robbie over email. We put our Twitter handles in there. Feel free to reach out over Twitter too. And we're more than happy to work with you. And just want to reiterate, this is just the beginning of collaboration that we hope to start between pre-medical students and engineers and SIR. So we're definitely going to have more events down the road. Just keep an eye out. All right. So with that, we will end our session for today. And again, thank you all for joining.
Once again, thank you, everybody. I hope you had a nice time. Thank you for giving up some time on your Saturday.
Thanks, everyone.